So hello guys, welcome to my YouTube channel Med School with Shubham. I am Shubham, a third professional MBBS student, and I shall be teaching you anatomy. I have started a new series on embryology on my YouTube channel in which I have completed the first week of development. Today we will be starting with the second week of development. In the second week of development, we will be learning about the changes that will be taking place in the embryoblast, the changes in the trophoblast, and the changes in the endometrium. This is part 1 of the second week of development, and in today's video we will be learning about the changes that are going to take place in the embryoblast in the second week of development. So till the last lecture we learned that the blastocyst contained the trophoblast and the embryoblast. The trophoblast further got differentiated into cytotrophoblast and syncytotrophoblast. The one which contained the cell membrane was known as the cytotrophoblast, and the one which does not contain the cell membrane is called as the syncytotrophoblast. Now we have the embryoblast. We have to learn about the embryoblast. The embryoblast also gets differentiated into epiblast and hypoblast. These are two cells which are called as epiblast and hypoblast. So let's learn about epiblast and hypoblast in this diagrammatic form. So this was the blastocyst that we learned till the last lecture. This is the cytotrophoblast and this is the inner cell mass. Cytotrophoblast, and this is the embryoblast or the inner cell mass. Now this inner cell mass initially was in the form of a mass-like thing, that a bunch of cells, and now this bunch of cells got differentiated into a disc-like manner. Just assume that this is the mass of the embryoblast, they got differentiated into a disc-like form in this way, in which the disc has two layers. This is the upper layer, as you can see in this is the upper layer which is thick, and this is the lower layer which is thin. Similarly, the embryoblast also gets differentiated into a disc-like structure, and that disc shall be containing two layers, the lower layer, the lower one and the upper one. So the layer which is below and on the lower side, and the layer which is on the upper side. Okay, so let's name them. This layer which is on the lower side is called as the hypoblast, and the layer which is on the upper side is called as the epiblast. Hypo means below, epi means above, blast means cells. Now what will happen next? This epiblast and the hypoblast, the epiblast are columnar like cells, the hypoblast are squamous or lower cuboidal like cells. The epiblast is also called as primitive ectoderm, and the hypoblast is also called as primitive endoderm. Mark my words, these are the primitive ectoderm and primitive endoderm, and not the definitive ectoderm and definitive endoderm. Now the epiblast will now start proliferating, and it will cover this upper cavity. Let me show you how will this happen. This is the epiblast. This was the hypoblast, and this is the epiblast. Now this epiblast will start proliferating, and it will form cells which will line the upper cavity. So now these cells are called as the amniogenic cells, and this cavity which the amniogenic cells are lining is called as the amniotic cavity. Similarly, the hypoblast will also start proliferating, and it will cover the lower cavity in this form. Now these cells are called as the Usher's membrane. These cells are called as the Usher's membrane, and these Usher's membrane shall be lining a cavity which is below this one. This cavity is called as the primary yolk sac. Initially, this primary yolk sac, it was this cavity, which means this one. This cavity was initially known as the blastocele I hope you remember here. See, in this one, this was the inner cell mass, the outer cell layer, then zonal hatching took place and the uterine milk invaded inside, leading to, they pushed the inner cell mass towards one end of the embryo, forming this cavity which is called as the blastocele cavity, which contained this uterine milk. The blastocele is the same cavity that is the primary yolk sac. See, this is the blastocele cavity, this is the blastocele cavity, and now this blastocele cavity is now known as the primary yolk sac, and this primary yolk sac contains the same uterine milk, and the amniotic cavity contains a fluid which is called as the amniotic fluid. Amniotic cavity contains a fluid which is called as the amniotic fluid. So let's move forward. Now the cells of the primary yolk sac starts proliferating, and now the cells of the primary yolk sac, they will start proliferating and they will form a loosely arranged cell layer between the cytotrophoblast and the Users membrane. See, this was the cytotrophoblast, this is the Users membrane, and on this side we have the primary yolk sac. So the cells of the primary yolk sac, they will start proliferating and they will form a loosely arranged cell layer between the cytotrophoblast and the Users membrane. Now what will happen? A new cell layer has came between the cytotrophoblast and the Users membrane. So now they will start getting separated as a new cell layer is coming between them. Similarly, now this new cell layer, they will migrate upward, and they will migrate in this sense that they will now separate the cytotrophoblast and the amniogenic cells also. I hope you understand this. Let me explain you in the form of a diagram. See, this is the cytotrophoblast and this is the Users membrane. Here you have the primary yolk sac and the cells of the primary yolk sac, or some books even say that the cells of the primary yolk sac as well as the Users membrane. The cells of the primary yolk sac as well as the Users membrane, they starts proliferating, and now they will form a cell layer between the cytotrophoblast and this Users membrane in this form, and this cell layer will now migrate upward in this direction, and now they will form a cell layer between the amniogenic cells and the cytotrophoblast. Now what will happen? This amniogenic cell, Users membrane, amniotic cavity and the primary yolk sac, they are now in contact with the cytotrophoblast. Because of the formation of this new cell layer between all of these, they will get isolated and the size of embryo will also increase. So it will look something in this form. Epiblast, these are the epiblast, this is the amniogenic cells, this is the amniotic cavity, this is the hypoblast, the Users membrane and the primary yolk sac. I have drawn in the form of line and not the exact cells. Now what will happen? A new cell layer is now appears between this layer, between this space, in this way. All over you can find this new cell layer. Now let me name this thing. See what will happen. New cells will be formed, released in this manner, and now these cells will also migrate upward and they will separate this thing. I hope you understand this thing. Let me name this new cell layer. This is called as the, see guys, this thing is the actual embryo. This is going to form the actual baby. So this is the actual embryo of this entire thing, and this layer is outside the embryo. Therefore it is called as the extra embryonic mesoderm. I hope you understand why extra embryonic, because This is the actual embryo of this structure. Means from this the baby will be formed. This epiblast and the hypoblast, the baby will be formed, and everything apart from that is something outside the embryo. Therefore, it is called as the extra embryonic mesoderm. Now, what will happen in the next step? Small cavities will start be forming in this extra embryonic mesoderm, and now all these cavities will now merge together to form a large cavity, which is also called as a coelome, and this coelome is formed in the extra embryonic mesoderm. Therefore, we can call that as the extra embryonic coelome. Epiblast, amniogenic cells, epiblast, amniogenic cells. Similarly, hypoblast and Users membrane, amniotic cavity, primary yolk sac. And now extra embryonic mesoderm. Okay, so this thing is called as the extra embryonic coelome. Extra embryonic coelome. Why? Because this coelome is formed in the extra embryonic mesoderm. Therefore, it is called as the extra embryonic coelome. Now, because of the formation of the extra embryonic coelome, the extra embryonic mesoderm gets differentiated into two parts or two types. The one which is in contact with the primary yolk sac, that part of extra embryonic mesoderm which is in see, this is also extra embryonic mesoderm, this is also extra embryonic mesoderm, this is also extra embryonic mesoderm. All over you have the extra embryonic mesoderm. But that part of extra embryonic mesoderm which is in contact with the primary yolk sac and the Users membrane, that part is called as the splanchnopleuric extra embryonic mesoderm, and that part of extra embryonic mesoderm which is in contact with the cytotrophoblast. See, this is the cytotrophoblast. So that part of extra embryonic mesoderm which is in contact with cytotrophoblast as well as the amniogenic cells. Here we have the amniogenic cells. So that part of the mesoderm which is in contact with the cytotrophoblast and the amniogenic cells. So that extra embryonic mesoderm is called as the somatopleuric extra embryonic mesoderm. Splanchnopleuric extra embryonic mesoderm and somatopleuric extra embryonic mesoderm. Now you can also see there is a part where the extra embryonic coelome is absent. This one. In this part the extra embryonic coelome is absent. So this part of the extra embryonic mesoderm where the extra embryonic coelome is absent is called as the undifferentiated extra embryonic mesoderm. Undifferentiated extra embryonic mesoderm. Okay. So let's move forward. Let me revise everything once again in this manner. Okay. So this was the epiblast. This is the embryoblast. Embryoblast gave rise to two cell layers, the epiblast and the hypoblast. The epiblast were columnar cells, hypoblast were squamous or cuboidal-like cells, lower cuboidal-like cells. Epiblast is also known as the primitive ectoderm, and hypoblast is also called as the primitive endoderm. The primitive ectoderm and primitive endoderm, both of these are germ layers. Therefore, we call second week of development as the week of the bilaminar germ disc formation. Bi means two, laminar means layer, germ disc This is in the form of a disc, therefore germ disc formation. Germ, why germ? Because ectoderm and endoderm are involved. Both of them are germ layers, therefore bilaminar germ disc formation. Now epiblast will give rise to amniogenic cells, hypoblast will give rise to the Users membrane, amniogenic cells will line the amniotic cavity, Users membrane will line the primary yolk sac. Now the Users membrane and the primary yolk sac will give rise to a new cell layer which is called as the extraembryonic mesoderm. Small cavities will start forming inside this extraembryonic mesoderm, and that is called as the extraembryon. All these cavities will now merge together to form a big coelome, and this coelome is called as the extraembryonic coelome. Because of the formation of the extraembryonic coelome, the extraembryonic mesoderm is now gets divided into two types, the splanchnopleuric extraembryonic mesoderm and the somatopleuric extraembryonic mesoderm. The one which is in contact with the primary yolk sac is called as the splanchnopleuric extraembryonic mesoderm, and the one which is in contact with the amniotic cavity and the cytotrophoblast is called as the somatopleuric extraembryonic mesoderm. Let's move forward. Now what will happen? A small part from this primary yolk sac will get pinched off or will start getting separated, and this will finally get separated. And now this part of the primary yolk sac which got separated from the primary yolk sac is called as the exocoelomic cyst. And now after this exocoelomic cyst has got separated, the primary yolk sac is now named as the secondary yolk sac. Now this exocoelomic cyst will now get dissolved. The exocoelomic cyst will get dissolved in the extraembryonic mesoderm. It will get dissolved in the extraembryonic mesoderm. All these things I have named the dates, I have mentioned the dates of all these events. So the epiblast and hypoblast formation takes place on the day 8. Now the amniogenic cells and Users membrane takes place on the day 9 of development. The extraembryonic mesoderm is formed on the day 10th to day 11th. On the day 12th we can find the formation of the somatopleuric and the splanchnopleuric extraembryonic mesoderm and the extraembryonic coelome. On the day 13, the small part from this primary yolk sac starts getting pinched off or separated. This pinched off structure is called as the exocoelomic cyst. And on the day 14, the exocoelomic cyst gets separated out from the and gets dissolved in the extraembryonic mesoderm. Now, two important things we have to learn now. Let me focus on this thing. This was the thing which we saw till here. Now we will take the same diagram over here and let's focus on these structures. I hope this black color, you know what is this? This is the cytotrophoblast, and this yellow color one is the somatopleuric extraembryonic mesoderm. Cytotrophoblast plus somatopleuric extraembryonic mesoderm, both of them together is called as the chorion. The cytotrophoblast and the somatopleuric extraembryonic mesoderm, both of them together is called as chorion. Now let me emphasize on this structure. The amniogenic cells and the somatopleuric extraembryonic mesoderm. The amniogenic cells and the somatopleuric extraembryonic mesoderm, both of them together we call it as the amnion. The chorion and the amnion. Now this chorion and amnion are very important structures that we shall be learning in the upcoming lectures. But this is the base or the understanding of what actually is chorion and what actually is amnion. Many students don't actually understand from where does chorion comes, from where does amnion comes, what is somatopleuric extraembryonic mesoderm, what are the amniogenic cells. They just learn these things, ratify all these things, and then they forget everything. Now I have shown you in the form of a flow chart manner. So now you in a form of a story. So now you won't forget this in your entire life. Now this becomes very easy for you to learn the concepts and all these things. I hope this is very clear to you. See, one more thing, this chorion you can find all over, because everywhere you will find the cytotrophoblast and the somatopleuric extraembryonic mesoderm. So if this is the entire embryo, so everything covered is the chorion thing, because everything is the cytotrophoblast like this, and below the cytotrophoblast you have the somatopleuric extraembryonic mesoderm. So the embryo is outsidely covered with the chorion layer. Collectively it is the chorion layer. What is the use of the chorion layer? What is the use of the amnion layer? We will be learning in the upcoming lectures. Or you can say what is the significance of chorion and amnion? We shall be learning in the upcoming lectures. Now let's come to another important thing. As in my every lecture I focus more on the clinical anatomy. So in this lecture also we will be learning about the clinical anatomy. So today the clinical anatomy that we shall be learning in this lecture is the amniocentesis process. What is amniocentesis process? As I said that this is the amniotic cavity lined by the amniogenic cells. This amniotic cavity, it contains a fluid which is called as the amniotic fluid. Amniotic cavity contains a fluid which is called as the amniotic fluid. Now what does doctors do? They extract some amount of the amniotic fluid from the amniotic cavity and they send this amniotic fluid for some clinical tests. Now there are some cells inside this amniotic fluid, and these cells are now been gone through some clinical tests and gene testing, and now any genetic defects if there is present in the baby, that can be diagnosed in the in the embryonic stage only. So we can write that what is amniocentesis? The amniotic fluid is extracted from amniotic cavity. Now this amniotic fluid is sent for clinical test. And what is the significance of this process? The diagnosis of the genetic defects. Diagnosis of the genetic defects in the baby. Or in the embryo or in the fetus, whatever you can say. Right, in the embryonic stage you can find that whether the baby has any genetic defects such as Down syndrome, Klinefelter syndrome, or the Turner syndrome, or even sickle cell anemia, thalassemia. You can find out in the very embryonic stage, and if possible you can correct that also by any kind of gene expert treatment. Still that are going under trials, but it has not came into picture nowadays, but some trials are going on that thing, that how can the genetic defects be treated right in the embryonic stage. So what is the next thing? In the same thing, same process of amniocentesis, it can also be used for determining the estimation of the level of the alpha fetoprotein. Alpha fetoproteins. Now these alpha fetoproteins are also present inside this amniotic fluid. If the levels of the alpha fetoproteins are high, then there are chances that the baby can develop neural tube defects in the future. Therefore, this can also be diagnosed in the early stage. So this is about the clinical anatomy of these things. So today I have completed the changes that are going to take place in the embryoblast in the second week of development. I hope I was very clear in explaining you all the concepts of the second week of development. What is epiblast, what is hypoblast, what is extraembryonic mesoderm, what is extraembryonic coelome, what is amnion, and what is chorion. All these things are very important for you to understand the systemic embryology further. Therefore, general embryology actually forms the base of the systemic embryology. So thank you guys. Thank you for being with me till this point. Like my videos, share my videos, and also subscribe to my YouTube channel. Thank you so much guys.